I'll be explaining Nuremberg and Mattei's experiment dealing with genetic code. So before the experiment began, they needed to create a synthetic RNA for the experiment, a homopolymer. So in this case, they took uracil nucleotides and polynucleotide phosphorylase. The good thing about polynucleotide phosphorylase is that it doesn't require a template and it binds to any RNA nucleotide. And here, a poly-U homopolymer was created for the experiment. So the homopolymer was added to a test tube, 20 test tubes actually. 19 of them were unlabeled amino acids, and one was a radioactive amino acid. So here the tube was incubated, translation took place, the protein precipitated, condensed to the bottom of the tube, which helped free amino acids. Then it was filtered out and checked for radioactivity. Now steps 1 through 4 were repeated 20 times, and each tube was different amino acid was used, and the same thing, 19 unlabeled and then 1 radioactively labeled. At the end of the experiment, you have the 20 tubes listed here, and the tube with the protein that was radioactively labeled had a newly synthesized protein. In this case, phenylalanine was that synthesized protein. Now here they could determine that the codon for phenylalanine is UUU, triple U. So here, this experiment was important because they found a way to develop a method to identifying amino acids using a homopolymer. So here they would identify many and find many other amino acids, codons. So the other experiment I want to talk about is Nuremberg and Letter's experiment. So in this experiment, they synthesized 50 short RNAs with known codons and added them to individually to a mixture of ribosomes and tRNAs. Here, they were able to isolate the tRNAs that were bounded to mRNAs and ribosomes and determine which amino acids were present on the bounded tRNAs. And the important of this experiment was they were able to find other codons and determine them and label them by using this specific method.